Eukaryotic cells have a nucleus which contains chromosomes, which are made up of long strands of DNA that contain genetic information. In eukaryotic cells, there are chromosomes, which are long strands of packaged DNA molecules. These are also known as genes. Humans have 46 chromosomes in total, for 23 from each parent. 22 of these pairs are autosomal, and one pair is the sex chromosome, which determines the biological sex of the offspring. Chromosomes have a way of compacting so they are able to fit within the nucleus. This way of compaction will also ensure that the chromosomes are organized so they can be dealt out to the daughter cells during mitosis. In the interphase stage of mitosis, chromosomes are at their loosest state, but they are still organized in such a way within the nucleus to avoid the chromosomes from being tangled. One of the first levels of compaction involve histone proteins, which are the main components of nucleosomes. There are four histone proteins which are a part of the nucleosome: H2A, H2B, H3, and H4. H1 will come into play later. Two copies of each of the four proteins come together to form an octamer, which is in the shape of a spool. DNA will then wrap around the histone spool in around 147 base pairs in length, and it will help to compact the DNA. This forms a nucleosome, the histone octamer with the DNA around it. There is linked DNA between a nucleosome, which keeps them together and forms long chains. This will end up forming a 30 nanometer fiber, which is the least packaged form of DNA. It is a coiled helix of nucleosomal DNA. H1 is the linker histone is needed here in order to bind to the edge of the nucleosome to change the path of the DNA to promote the coiling for the 30 nanometer fiber. Like most things in the human body, chromosomal structures have to be regulated, and this is done through HATs and HDACs. HATs are histone acetyltransferases, acetylates histones, which loosens nucleosomes and allows access to DNA, and therefore allows transcription. HDACs, or histone deacetylation complexes, deacetylate histones, which lock the nucleosomes and prevents access to DNA, and therefore prevents transcription. The process of DNA replication allows for DNA genomes to be copied with few mistakes made. If there are mistakes made, DNA repair will find the mutations, isolate them, and fix them by any means. Replication begins at the origin of replication, where initiator protein binds and pries the two strands of DNA apart by breaking the hydrogen bonds between the bases. This creates a replication bubble. In the replication bubble, there is the replication fork, which is the site of active DNA replication. It is a junction where double-stranded DNA is separated and new strands are made using the old as a template. DNA is bidirectional since the replication fork forms on either side of the replication bubble and moves away from each other in opposite directions. There is an enzyme called DNA polymerase, which is one of the most important in replication. It makes a new DNA strand by using an old one as the template. DNA polymerase makes a phosphodiester bond between the 5' phosphate of an incoming nucleotide and the 3' hydroxyl on the end of the newly synthesized DNA strand. Deoxynucleotide triphosphates then enter the reaction site of the DNA polymerase. These are DNTPs, which are DHTP, DGTP, DCTP, and DTTP. The rate of DNA polymerization is sped up by the use of a clamp protein that encircles the DNA and interacts with the DNA polymerase so that the enzyme does not float. This process requires an additional protein called the clamp loader, which puts the clamp on the DNA. The replication fork is asymmetrical, as only one strand is able to be synthesized continuously. The bottom strand of the replication fork can be synthesized continuously because it starts synthesis at the end of the template and makes new DNA from the 5' end to the 3' end. The top strand, or the lagging strand, cannot be synthesized continuously. In the top strand, DNA has to be synthesized moving away from the replication fork, since DNA has to be made from 5' to 3'. The fragments of DNA that result from the lagging strand are called Okazaki fragments. These fragments are eventually sealed together using three enzymes. The first enzyme lands on the 3' end of one fragment and consumes the RNA primer. Then a repair polymerase puts down new DNA in place of the primer that was just consumed. Finally, DNA ligase seals the backbone of the DNA fragments to create a long continuous strand. DNA polymerase is one of the most important aspects of replication, but it makes many mistakes in the nucleus. In order to fix this, it checks each nucleotide that enters the reaction site and makes sure that the nucleotide is correct and fits with the base that is across from it in the template strand. But if it does let in the wrong nucleotide, it can fix it. DNA polymerase has a method called proofreading to ensure that the mistakes are fixed. When the DNA polymerase can feel that the nucleotide is not correct, since the DNA is no longer smooth, it shifts the incorrect nucleotide to a different location where the phosphodiester bond is cut, which then cuts off the incorrect nucleotide. DNA helicases are the enzymes that pull the DNA strands apart, and they are one of the most important enzymes in DNA replication. In order to pull apart the DNA strands, they have to use ATP in order to spin fast enough to accomplish this task. Single-stranded binding proteins keep the single-stranded DNA separated from each other. Telomerase is an enzyme that is attracted to the ends of chromosomes and makes telomeres. This enzyme carries its own template for RNA to make short DNA sequences. It does this at the end of a chromosome, so a large cap is made that contains an abundance of DNA repeats in tandem. Telomeres eliminate the shortening effect that would occur during each round of replication. Every so often there is a permanent change to the DNA sequence that does not get repaired during polymerase proofreading, and this change is called a mutation. Mutations can have profound and devastating effects if they occur in vital regions of DNA sequences. Even a mutation like a base substitution where a cytosine is switched with a guanine can have drastic consequences. There are two classes of mutations. One is somatic mutations, which occur in any cell that is not the gametes, which are the sex cells. The other is germline mutation, which occurs in the gametes. This mutation can be passed on to the next generation. When a mutation does occur, the cell has a few systems it uses in order to try and fix the mutation. One of these systems is called the mismatch repair system. This system will excise a section of the new DNA that includes the mismatch, and then the old strand of DNA is used as a template to fix this error. In bacterial cells, they have a process called methylation, which marks the old DNA. This methylated strand is used in order to correct the new DNA strand so it matches the correct one. In eukaryotes like humans, it is not known how the DNA strands are distinguished as old and new. The repair system is the best defense against cancer and somatic mutations. There are two more repair systems. One of them is base excision repair. In this repair system, a mutant base is recognized and excised out of the DNA. DNA polymerase then fills the single nucleotide gap, and it is then sealed by DNA ligase. The second repair system is called nucleotide excision repair. In this repair system, when a distortion is found, the DNA strands in that region are separated. The DNA is severed on both sides of the mutation, which leaves a gap. It is then filled by DNA polymerase and sealed by DNA ligase. There is a potentially lethal mutation that can occur in cells which is called a double-stranded break. This mutation completely severs DNA molecules in two pieces. There are two recombination processes that can fix this mutation: homologous and non-homologous recombination. The first mechanism is called non-homologous recombination. In this process, the two broken ends of the DNA molecule are blunted and then ligated together. Despite being repaired, nucleotides are lost in the blunting process. The second mechanism is homologous recombination. In this process, the information on a similar close DNA is used as a template for making the repair. While not identical, it is close enough for the repair to occur. This process begins when a nuclease consumes some of the nucleotides on one of the strands at a double-stranded break. The single-stranded broken end of the DNA checks the nearby DNA molecule to ensure there is similarity before proceeding. If they are similar enough, strand invasion will then occur and it creates a branch point where the two DNA strands from both molecules cross. The invading strand is elongated by a repair polymerase. The branch point migrates to allow additional DNA to be made when the DNA is synthesized. Finally, the invading strands are nicked to allow them to return to their regular positions, and DNA ligase seals the nicks. This forms two completely repaired DNA molecules. In eukaryotic cells, the start of transcription begins with a promoter called the TATA box. There are three RNA polymerases that are involved. RNA polymerase I and RNA polymerase III transcribe genes that code for transfer RNA, ribosomal RNA, and small RNAs that are important for the cell structure and function. RNA polymerase II transcribes a majority of eukaryotic genes, such as those that code for proteins. RNA polymerase also needs general transcription factors that have to assemble at the promoters with the polymers before it can begin transcription. General transcription factors are accessory proteins that assemble on the promoter in order to position the RNA polymerase, separate the double helix, and launch the RNA polymerase for transcription. The GTFs usually assemble at the promoter that RNA polymerase II uses. The assembly process begins with transcription factor 2D binds to a short DNA sequence that is composed of T and A nucleotides. This sequence is known as the TATA sequence or TATA box. When transcription factor 2 binds to the DNA, it causes a distortion to serve as a landmark for the other assembly proteins. The TATA binding protein binds to the TATA box with transcription factor 2D. Then transcription factor 2B binds at the site as well. Then transcription factor 2E, transcription factor 2H, and transcription factor 2F also bind at the location, where then UTP, ATP, CTP, and G2P is then used to create transcription. RNA polymerase II has a tail which is an end of its amino acid chain that is accessible and modified. This is the end of the amino acid chain and is called the carboxyl terminal end. When this tail is phosphorylated, it serves as a polymerase code for RNA that is similar in function to the histone code. The tail is phosphorylated at a specific region to signal the release from the promoter. The transcription factor 2H does this. After RNA polymerase II has begun transcribing, most of the GTFs leave the promoter. mRNA has to be processed first before it can be exported into the cytoplasm for translation. First, mRNAs receive a 5' cap, which is a methylated guanine attached in an odd way. This is added to the 5' end of the mRNA. It is the first marker that marks the mRNA as genuine. Next, it receives a poly-A tail, which is hundreds of adenines long. This is added to the 3' end of the mRNA. It marks the end of the mRNA as genuine. The two additions mark the full mRNA as genuine and make it more stable so it can exit the nucleus and be translated. Last but not least, the proteins recruited to the 5' cap and the poly A tail interact, which causes the mRNA to circularize. mRNA has both exons, which are codon sequences, and introns, which are non-codon sequences. The introns are not essential to the mRNA and has to be removed. This is done through a process called splicing. In this process, a protein machine called spliceosome removes the introns and ligates the exons together. Finally, when the mRNA is ready to leave the nucleus, it has to go through the nuclear pore complexes, which only allow mRNA out of the nucleus if they are bound by proteins at the splice junctions, tail, and cap. In alternative splicing, not all of the exons have to be kept. New proteins can be made by deleting some exons in between when splicing non-adjacent exons together. About 60% of human genes will undergo alternative splicing because many different proteins can be made from one gene in this process. A codon is a set of 3 nucleotides that will be translated into an amino acid. 3 nucleotides are needed to code for one amino acid, and so there are 64 amino acids despite only needing 20. This allows for more than one set of triplets to code for one amino acid, which helps decrease the amount of mutations that occur that have drastic effects. Like any other sequence that needs to be read, there are different ways of reading them. But for nucleotide sequences, the right reading frame has to start with an AUG, which codes for start, and then ends with a stop codon. This kind of reading frame is called the open reading frame. Ribosomes are collections of RNA and protein molecules that form a complex that performs protein synthesis. There are 2 subunits in the ribosome. The smaller one matches the mRNA codons to the proper tRNA, and the larger forms a peptide bond between amino acid which links them together in a chain. The subunits come together with mRNA in the middle of them. The codons in mRNA cannot bind to the corresponding amino acids, and this is where tRNA comes into play. They have anticodons on one end, which base pairs with the codon of the mRNA. Then they carry the corresponding amino acid on the other end. The tRNA essentially serves as a translator because it can translate both codons and amino acids. There are 4 regions on base pairing in tRNA, which gives it a clover leaf structure, which then folds in half so that the important parts of tRNA are exposed and inaccessible. Unlike amino acids, there are not 64 tRNAs. There are only 31 different tRNAs, but most can code for multiple codons through wobbling, which makes it possible for them to recognize multiple codons. Before it can be translated, tRNA must be charged with amino acid by aminosyl tRNA synthetases. This process is called charging, and it is how an amino acid is linked onto the corresponding tRNA. Once this is completed, translation can finally begin. In translation, first a tRNA with the right anticodon and appropriate amino acid enters the A site in the ribosome, which binds to the codon. The amino acid on the tRNA then forms a peptide bond with the amino acid chain that's linked to the tRNA in the P site. It transfers that amino acid chain to that tRNA. The ribosome then shifts forward by one codon, and the tRNAs stay base paired with the codons. This process is called translocation and moves the tRNA from the P site to the E site. Translation has to have a starting point and an ending point, which is why a specific tRNA is needed to start translation. This tRNA is called the initiator tRNA, which associates with a small ribosomal unit which binds to the 5' end of an mRNA. It keeps the mRNA engaged while the initiator tRNA scans for the start codon. When the start codon is found, the initiator tRNA's base paired to it and their larger subunit joins the complex. Stop codons terminate translation, and this is done when a stop codon is positioned under the A site. Release factors then enter the A site and bind to it. They catalyze the release of the amino acid chain from the tRNA in the P site, which releases it from the ribosome. In eukaryotes, transcription factors combine to enhancers, which can be thousands of base pairs away from the target gene. It means that there will be regulatory sequences in the gene scattered across entire chromosomes and have unrelated DNA sequences between them. Most of our genes must be turned off or on by multiple proteins working together. This is called combinatorial control, and this is how our genes are controlled. Activators in eukaryotes aid in transcription initiation and do not affect RNA polymerase binding to the promoter. An important protein complex is called the mediator, which is found on the end of RNA polymerase II. The mediator receives the signals that affect the gene, integrate them, and pass them on to RNA polymerase II. RNA interference is used by cells for silencing genes, which requires the formation of double-stranded RNA. Once it recognizes double-stranded RNA, all double and single-stranded RNAs with the same sequence are destroyed. RNA interference is not found in prokaryotes, but it is found in most eukaryotes. It is used in laboratories to prevent expression of targeted genes.